I want to talk a little bit about the regulatory landscape.
Okay.
What are some of the key regulatory uncertainties or needs in the EV field, and how are your regulatory teams with Inside Egley working to anticipate and navigate these, especially, of course, in the United States?
Yeah, so I think one of the challenges always with any kind of biologic product is a potency assay. And even though the FDA has come out with a number of guidances on potency assays for biologics and for cell and gene therapies, they're pretty vague. They don't tell you a lot of what you actually need. And when you actually go to the agency with these proposals, they typically want, because there's not a concrete one mechanism of action, this is the target where you're targeting, this is the receptor, and here, look at how great this targets the receptor. There's not that here. And so they often want a matrix of assays that are sort of orthogonal and that all point to the same answer. They also want them to be stability indicating. For potency, typically they want them to be stability indicating. If that's confusing, what I mean by that is that if you have product that you just normally make and it's working as expected, it falls in a certain range in your potency assay. If you quote unquote break the product, right, you damage it in some way so that it's no longer has that function, then it shows that, right? It fails it. Your assays fail it. So it would indicate that there's something wrong with it. That is harder than it sounds, right? It seems like you should be able to boil it, drive over it, break it in some way, and have it read out on your potency assay. It's not always a perfect connection between those two. And that can get pretty problematic when you're trying to say, yeah, this is a great potency assay, and yet you can't always say in all cases where you know that it's no longer functional, assuming you have an appropriate assay for that. Some of those assays are pretty crude, unfortunately. So if you have an assay for wound healing, for example, wound models in vivo are not very good, right? And trying to model it in vitro, is almost impossible. And we don't have a lot of good translational models. I mean, sure, there are some. And when I say they're not good, they're fine. All due respect to the people who developed those models in pigs and rats and others. What I mean by not good in this setting is that they're not necessarily predictive of how that product is going to do in the human setting. And lots of our models suffer from that. That's not new in this field. But that's kind of what we're relying on, right, to prove that something is stability indicating or not. So if you can find a good set of potency assays and find a way to say that they're stability indicating, that's a huge win. And that's something that I would really encourage people, even though they hate doing it, encourage people to start early on, even though you think it's way too early. We just want to see if this works, is often what I hear. But if you can't find a way to show whether something's active and potent, then it's going to be very difficult to move fast and furious. The one thing, so you asked what we're doing, some of the things that we're doing, we are focusing our potency assays on things on the indications. So if you look at wounds, it's biologic, autologous, and allogeneic biologics. And by biologics, I'm talking about anything from monoclonals to autologous cell therapy, or Polarity TE has skin that they use to treat other skin on the same person. And there's so all of those sort of mixes of things, PRP. I mean, so you run the whole gamut of things that I would call biologics. And it's pretty well established in the agency for wounds what you would look at for that. So that's where we've headed. And that was for burns and EV. Now, if we're going to look at graft versus host, we have to look at a different type of potency assay because those aren't going to work as well. And if you think about that, that can be pretty onerous, right? So if you're a company that wants to have a robust pipeline that's very different, and you want to do hepatic disease, and you want to do some sort of another immune-mediated disease, you want to do some, lung, you want to get a lung, well, that's going to be really difficult to do all those potency assays. So that strategy is probably not good if that's what you're trying to do. Because we right now are focused on sort of local type delivery of things we can get to in that space, it's a little bit easier for us. I'm not saying that strategy works for everyone, but that's where our approach is.
Great. Okay, thank you. And I definitely want to reiterate what you said about starting early. I think from every professional and executive that we've spoken to, certainly with regard to regulatory, also manufacturing, but really what have you within the life cycle of the therapy, thinking with the end in mind and starting as whatever that thing is, starting as soon as possible and as early as possible. So um, echoed sentiments from here as well. I want to talk a little bit about the competitive landscape from a commercial strategy standpoint, perhaps market adoption, um, for exosome therapies in dermatology and regenerative medicine. What does what does that landscape look like, and how is Egle positioning itself to differentiate?
Mm. Yeah, so I was just recently been looking at some of this, and like I said, there's over 70 known, I'm sure there are some stealth ones as well, 70 known companies around the world that are mostly in the US and Europe that are trying to develop companies, entities. Some of them are coming out of academic labs and moving into the VC and angel world, that are developing EV products. Not too many of them are in dermatologic applications. There's a wide range of indications. So, autoimmune, immuno-oncology, inflammatory, systemic inflammatory diseases. There's, as I said, there's quite a bit of interest in neural stem cell EVs or exosomes for any number of either rare neurologic diseases or neurodegenerative diseases, or things like post-stroke or traumatic brain injury. So there's quite a bit of that going on now, given the availability of the blood-brain barrier passage. So those aren't directly competitive with us at this time, and most are. And I believe, and I might be wrong about this, but based on what I know, we're one of the few that are actually at clinical stage. There's a handful that are definitely ready or coming on at clinical, and the majority of those are not in our space. So I think right now that's not as kind of what keeps us up at night as things like manufacturing, right? Is how we compete in the market. But we do have, when that occurs, and when and if we do have that sort of competitive pressure, we do have a unique way of manufacturing that actually was sort of serendipitous with our CSO, where he was working out in his lab at the University of Miami. They were working out ways to make these things in the lab setting. And the typical way that they were being made, they weren't doing as well as this other way that they were making them in their porcine wound studies. And he actually did a study I believe it's been published, a study comparing the old way to the new way, and found that there was a dramatic difference in how they performed. So I think, at least in that sense, we do have a differentiation there. And we also differentiate in that not that many people are using all of the extracellular vesicles. They're typically using exosomes. It's more uncommon to have the EV-based therapeutic.